Hi everyone, welcome to Chapter 21, our final lecture of the semester, which covers the topic of viruses. Viruses are something that we have all encountered in our everyday lives. But despite this familiarity, we often don't fully understand what viruses are and how they work. And so to start this lecture off, I want to invite you to imagine a scenario that many of us may be familiar with. Let's say that you have a fever and a sore throat, and you think that you might have strep throat, which is a bacterial infection. So you go to the doctor, and the doctor assesses you. They take a cotton swab, they stick it in your mouth, and they rub it up against your tonsil to collect a sample. And they pull it out, and they take that swab, and they run a test on it there in the doctor's office to determine whether or not you have strep throat. And when the test comes back, it's negative. So your doctor tells you, well, it looks like you don't have strep throat. You must just have a virus. So I can't give you any antibiotics. You're just going to have to go home and fight this infection off on your own steam. Many of us have probably experienced this before, being told that you have a virus after you go to the doctor. And we might leave a little bit confused about what is the nature of this infection that can't be treated with antibiotics? What is it that a virus does to my body? So there are many misconceptions about what viruses are and what they do. And we're going to start off by actually laying down a few facts about what viruses are not. Viruses are not bacteria. These are two separate classifications of biological entities. Bacteria, as we've previously learned in this class, are single-celled, microscopic prokaryotic organisms, and while some of them cause infections, most of them do not. Viruses, on the other hand, are all infectious. All viruses are parasites that need to infect cellular organisms in order to survive. Viruses are also not made of cells, and this also distinguishes them from bacteria. They are made of proteins and nucleic acids, and we'll go into more detail about this in future slides, but they are acellular. They're not made of cells. Viruses are not visible to the naked eye. They are extremely tiny, and furthermore, they are extremely tiny even compared to bacteria. In fact, there are viruses that actually infect bacteria, and many hundreds of thousands of viruses can fit inside of the volume of even some of the smallest living cells. And then lastly, this one's a really weird one. Viruses are not alive. They are not considered by biologists to be living organisms. And we'll talk about why this is the case in a few slides down the road as well. But just to briefly give you a picture of how small viruses are compared to living cells, here we have shown some of the smallest viruses and then gradually getting larger in size all the way up to a larger virus like Ebola here. And then over here, we have certain types of cells. We have a chlamydia bacteria cell. We have an E. coli bacterium, this large oval-shaped one, and then this human red blood cell that is totally dwarfing everything else. And so you can see how small these little viral particles are compared even to bacteria. And bacteria are the smallest types of cells that we know of. So we've laid down what viruses are not. So what are viruses? Viruses are parasitic infection-causing agents composed of nucleic acids. Remember, nucleic acids means DNA and RNA, enclosed in a protein coating called a capsid. So they have a very simple structure. It's genetic information, nucleic acids, enclosed in a protein. Now, the structure can take several different forms, even though they're all plays on this same format. Viruses can be helical in shape, meaning that they have a tube-like shape that contains their nucleic acid. So here we have the capsid represented as the tube, and then we have a helix nucleic acid coming up the middle. That's why it's called helical. They may also be icosahedral. An icosahedron is a very complex three-dimensional shape here. Under the microscope, it simply looks like a hexagon, but it is actually a 21-sided three-dimensional figure here, within which the nucleic acid is stored. Viruses can also have really strange-looking shapes that are referred to as complex. And in general, a complex virus is one that does not fit into either one of these two categories. If it's not helical and it's not icosahedral, it typically falls into this complex category. However, you can see that complex viruses can have certain pieces to them that resemble the two main categories, helical or icosahedral. For example, this virus right here, which is a bacteriophage, which we'll learn more about in just a second. This virus has part of its capsid is icosahedral in nature, and part of its capsid is helical in nature. But the general rule is that if it's not helical and it's not icosahedral, if it's not clearly one of those two, it is complex. Another component to the structure of viruses can be an envelope. And an envelope is a layer of lipids that resides outside of the virus's capsid. You may remember that cell membranes are composed of a phospholipid bilayer. And so these viral envelopes are somewhat similar in nature to a cell membrane, but they're not exactly the same. Not all viruses have an envelope, and ones that lack an envelope are called naked viruses. So in this figure right here, what we can see is that inside of the envelope, which is shown in yellow, there is the virus's capsid. In this case, we are looking at a helical capsid because it's tube-like in shape. And within that helical capsid, we can see the nucleic acid, the DNA or RNA shown in purple. We can also see on the surface of the envelope, there are these little green projections that are labeled as spikes. And spikes are especially important for viruses. They're very important for the ability of the virus to infect a host cell. So these spike proteins, which in this case are also shown in green right here in this figure, we have these little spikes projecting off the surface of the virus. What they do is they interact with receptors on the surface of a cell that the virus intends to infect and allow it to gain entry to that cell. So the spike proteins are sort of like the key that unlocks the door to a host cell. Those spike proteins can be present on the surface of the capsid if it's a naked virus, or if it's an enveloped virus, they will be present on the surface of the envelope. So this brings us to our first checkpoint. I have two electron microscopy images here of two different viruses, and I'd like you to indicate to me what is the capsid shape that we see in each of these cases. We've got Ebola virus on the left, and we've got smallpox virus on the right. So what is the capsid shape? And do these appear to be enveloped or naked in nature based on your best assessment? Now we're going to backtrack for a second because before we go on to talk more about viruses and the nature of the infections that they cause, we need to address a point that was made at the very beginning, which is that viruses aren't alive. Why are viruses not alive? Well, it's because they don't fulfill those 8 characteristics of living organisms that we learned about way back at the beginning of the semester. We talked about how there are 8 characteristics that all living organisms share. They, for example, are able to conduct metabolism, they have homeostasis, they are able to reproduce. And that one right there is the key to why viruses are not considered alive, because viruses cannot reproduce without help from a living cell. Viruses reproduce by invading the cells of a living organism, taking over that cell, and then using the cell's enzymes and resources to generate more viruses, which is what you can see going on In this microscopy image right here, each of these little hexagons right here, which are icosahedral viruses, these represent viruses that are reproducing inside of this host cell, but they would not be able to do so without the help of the host cell. Without a cell to infect, viruses are totally inert and cannot make more viruses, and this is why they are not considered alive. Viruses can infect all different types of hosts, although we most often hear about viruses infecting human hosts and how that applies to our lives. We may be familiar with certain viruses that can infect animal hosts, but it doesn't stop there. Viruses can infect bacteria. Remember, viruses and bacteria, not the same thing. Bacteria are prokaryotic cells, and there are viruses that can infect these cells. They are called bacteriophages. There are also viruses that can infect plants. Right here we see an electron microscope image of tobacco mosaic virus, which was actually the first virus ever identified which infects tobacco plants and also relatives of tobacco plants. And then, of course, there are animal viruses. Here on the screen, we see an image of coronavirus, the cause of COVID-19. These right here, this complex virus is rabies virus. And in our lecture, what we are going to look at is bacteriophages, viruses that infect bacteria. And we're going to look at a few different examples of animal viruses. Regardless of what type of cell a virus infects and what type of host it infects, there are a few basic steps that viruses carry out when they are doing this infection and replication process. The first step is called attachment, and this is where the virus attaches to the surface of the host cell using its spike proteins that we just talked about. The second step is entry. This is where the virus and or its genetic material enters the cell. Then comes replication and assembly. So the virus will hijack the host cell's enzymes and resources, and instead of the cell carrying out all of the processes that we have learned to support its metabolism, instead it redirects its energy to producing and assembling new viruses. And then finally, there is release. This is where the newly assembled viruses are released from the host cell, and then thereafter they can go on to infect new hosts. And sometimes this release process, as we will learn, occurs through the bursting and the death of the host cell, although other times it does not. So as I said, in this lecture, we are going to narrow our focus to a discussion of bacteriophages and animal viruses. We are first going to start by looking at bacteriophages. These are viruses that infect bacteria and bacteria exclusively. And as you can see, they look like weird little aliens. They have a complex shape that has an icosahedral head and a helical tail. They also have these tail fibers right here, and these are actually where their protein spikes are located. So these tail fibers interact with the surface of the host cell. Its genetic material is DNA, and that's found up here in the capsid head. And there are many different types of bacteriophages out there. And depending upon which type you're talking about, they have different life cycles that they carry out in order to replicate themselves. One of these two life cycles is called the lytic cycle. And I like to think of this as being like the hostile takeover version of the life cycle of a bacteriophage. In the lytic cycle, the bacteriophage invades the host cell, takes over, and then produces a large number of viruses rapidly until the host cell bursts and they're all released. So, hostile takeover. Other bacteriophages perform something called the lysogenic cycle. And the lysogenic cycle is more like a covert operation. In the lysogenic cycle, the bacteriophage invades a bacterium, inserts its DNA into the bacteria's chromosome, and then allows that DNA to lie latent while the host cell replicates. So let's take a look at each of these in a little bit more detail, starting with the lytic cycle. So as we can see, In this figure right here, we have the bacteriophage represented as this blue structure that has touched down on the surface of the bacterial cell, which is shown in orange. This purple thing is the bacteria's DNA, and this green thing is the virus's DNA. So bacteriophages will attach themselves to a bacterium, and then they will inject their DNA into the bacterial cell, kind of like a hypodermic needle. They will just contract, and that DNA will be passed into the cell. After this, the DNA will initiate a, like I said, hostile takeover of the host cell, and it will redirect the host cell to start creating new viral pieces and then assembling them into full and complete viruses. So it creates new capsids, which is the protein component. The cell also creates more viral DNA, and then the viral DNA is placed inside of the capsid, and eventually there are so many viruses inside of this bacterial cell, it bursts open and releases them all, which of course results in the death of the host cell. But this process of bursting open is called lysis, and this is the namesake of the lytic cycle. It's called the Ly lytic cycle because Ly lysis is what happens at the end of it, death of the host cell by bursting. In contrast, the lysogenic cycle, remember this is the covert operation version of the replication. It starts off the same way as the lytic cycle, where the bacteriophage attaches, injects its DNA, but once that DNA is inside of the bacterial cell, something called integration takes place. So instead of immediately taking over, like in the lytic cycle, that piece of viral DNA sneaks into the bacteria's chromosome and it hides in there. This means that every time the bacterial cell replicates, every time it divides and one cell becomes two, the bacterial cell does not realize that it has been invaded by this viral DNA segment. And so it will replicate the viral DNA along with the rest of its chromosome. And in this way, the virus sort of hitches a ride in a very sneaky manner to allow its DNA to be replicated generation after generation as a sort of hitchhiker in the bacteria's DNA. Now, this can go on indefinitely, but sometimes something happens which triggers that viral DNA to pop out of the bacterial DNA. Sort of the viral DNA, it gets discovered, it gets found out, and it exits the chromosome. This process is called induction. And once the viral DNA has been found out, once the hitchhiker has been discovered, then a hostile takeover happens. And the lysogenic cycle can actually turn into the lytic cycle. Replication and assembly will take place, and then eventually release via lysis. Now, the lysogenic cycle, because it integrates the viral DNA into the bacterial DNA, it can actually introduce new genes to the bacterium that it did not possess before it was infected with this virus. And relevant to us as humans is those genes can be harmful, and they can turn harmless bacteria into harmful infectious bacteria that have the ability to infect humans and cause harm to us. This phenomenon is called phage conversion. So conversion meaning a change, and phage meaning it's driven by the bacteriophage. So it's a change caused by this bacteriophage virus. Phage conversion is the introduction of new traits to bacteria that comes from a bacteriophage infection where the viral DNA introduces new genes. What do those genes look like? Well, I'll give you a few examples. There are a number of bacteria that, without having a bacteriophage infection, without having these genes, do not exhibit some of these harmful traits. For example, scarlet fever is a bacterial infection in humans, But the specific strain of the bacteria that causes scarlet fever is one that has been infected with a bacteriophage, and that bacteriophage has conferred a gene that produces a toxin that results in the characteristic rash of scarlet fever. Diphtheria, which is a disease that you don't see anymore in the United States because it's protected against by our normal vaccine schedule, but it is a highly deadly bacterial infection. There is a toxin in the bacteria that causes this disease, which originates from a viral infection of that bacteria. Bacteria of this variety that have not been infected by a bacteriophage are otherwise harmless to us. The same goes for cholera. Cholera is a highly deadly digestive infection, and the toxin that the bacteria that are involved in this infection generate is actually originally from a viral infection of that bacteria. So these are some examples of how phage conversion can introduce harmful characteristics to bacteria that would otherwise be harmless to us as humans. This is not to say, however, that bacteriophages are all bad, because there are bacteriophages that can also benefit us as humans by killing bacteria that are already harmful. And something called phage therapy is currently gaining a lot of attention for its potential to be sort of an alternative to antibiotics as a way to kill harmful bacteria and fight infections that are caused by bacteria. Because, as we learned, the lytic cycle is where a bacteriophage virus infects a bacteria, and at the end of the cycle, it ends with the death of that bacteria by bursting open. So I am going to show you this brief video here that is an example of an experimental phage therapy treatment that was used to treat a bacterial infection.
They were having a great time. They'd seen the pyramids and they were on a boat headed up to Luxor. And as the boat was pulling into Luxor, Stephanie texted me and said that Tom had developed abdominal pain and was unable to eat, and she was worried that he developed food poisoning. She then texted me back and said, the pain is going through to his back. Could this be pancreatitis? What should I do? And I said, you need to get him to a hospital. The last thing you need is pancreatitis in the middle of Egypt. It's an organism we have a lot of respect for now. So after a little over a week in Germany, Stephanie was able to work with people here to get him flown to UCSD. And he was hospitalized here at UC San Diego just before Christmas.
I wasn't ready to let you go. And I came to you and I held your hand and I said, honey, they're doing everything that they can do. There's nothing that can kill this bug, so if you want to fight, you need to fight. But do you want me to kind of pursue some alternative therapies, and we'll leave no stone unturned. Around that time, a colleague of ours who's at UCSF said that a friend of hers had been treated with phage therapy and flew to Tbilisi, Georgia, where she had this miraculous cure. And I thought, wow, that sounds kind of far out, but wait. I remember learning about phages when I was an undergrad at the University of Toronto. So I thought, wow, and I looked it up and I saw that it wasn't licensed and it certainly wasn't FDA approved, yet there were some papers saying that this was a promising area for the future. So I wrote Chip Schooley an email and I said, what do you think about phage therapy? And he said, what an interesting and intriguing idea. If you can help identify some phages that are going to be reactive against his isolate, I'm going to give it a whirl. We set on a phage hunt. Took just a couple weeks, but an international effort of researchers and people at the Navy Medical Research Unit came together to make this happen. We couldn't have done it without the UCSD team.
What they literally mean are bacteriophage. They eat bacteria. And these are small viruses that are ubiquitous. We're loaded with bacteriophage, and on any given day there's a dance going on in our GI tracts of bacteriophage attacking bacteria. In the early part of the 20th century, there were a lot of efforts in France and what became the former Soviet Union to use these organisms as therapeutic agents in people with difficult to treat infections. Toward the late 30s and early 40s, there was less interest in this in the West because antibiotics began to emerge, and people in the West thought antibiotics would be able to take care of our problems with bacterial infection. There had not been a lot of experience with treating people with bacteriophage, and almost none giving it intravenously in the U.S. So we relied on some of the literature from Soviet Union, but a lot of it was really being worked out kind of on a first patient basis as we went, using a combination of the previous literature, our own intuition about how these phage would circulate, and advice from people who'd been thinking about it for a long time. Within about 48 hours, he woke up and turned to his daughter and said, "I love you."
Had there been phages available in the course of my illness when I first got ill, I wouldn't have been nearly as sick as it turned out I was.
There's a lot of interest in bacteriophage therapy that's been gradually emerging over the last year or two as efforts to develop more traditional antibiotics have not really made the progress people had hoped. Bacteriophage have been kind of brought back off the shelf as a potential new approach to therapy. They're not simple to use. You have to develop a cocktail for each patient's own isolate. They seem to be relatively safe to give, but they're going to be difficult to develop from both the research perspective and also from the regulatory perspective. Now the good news is we have awfully good tools now to do this. Robotics and much more sophisticated molecular tools that enable this to be done. 10 or 15 years ago it would have been impossible to contemplate doing this. And so I think although there's a lot of research to be done, I think there are going to be a lot of clinical applications where this approach to therapy may be very beneficial to patients.
You can't emphasize enough how much, well, I believe, that phages really can change how we treat people in an economical way. So I see the third world and other places benefiting as much as we do. I mean, in my case, I can't tell you how much it costs to take care of me, much more than, I would like to believe. But if this can be done cheaply and really low-tech way, we can save millions of lives. And so it's, I think, the future of medicine.
So we are going to round off our discussion of bacteriophages with a couple of checkpoints about what we've just learned. Firstly, phage lambda, that's the Greek letter lambda. Phage lambda is a bacteriophage that infects cells of E. coli, which is a bacterium that lives in your digestive tract. It integrates its DNA into the E. coli cell's DNA. Which life cycle is exhibited by phage lambda? And secondly, would phage lambda be a good virus to use for phage therapy? Why or why not? So now that we've discussed bacteriophages, we are going to move on to look at some different animal viruses. And we are going to focus on just a select few examples that sort of emblematize the different variations that we can see across animal viruses that infect humans. Those three are the influenza virus, coronavirus, the cause of COVID-19, and human immunodeficiency virus, or HIV. So we'll move through these one by one, starting with influenza virus. The influenza virus is an enveloped virus, shown here in this cartoon depiction at the right. Its nucleic acid that it contains within its capsid is RNA, not DNA. So viruses are unique in the biological world in that they can have RNA as their genetic material rather than DNA. The influenza virus has two different types of spikes on the surface of its envelope. It has what are called H spikes, named for the fact that they're composed of a protein called hemagglutinin. And these hemagglutinin spikes play the role of interacting with the receptors on the surface of host cells and gaining entry. But the flu virus also has N spikes, which are named for the fact that they're composed of a protein called neuraminidase. And these N spikes actually play sort of the opposite role of the H spikes. They send signals to the host cell to mediate exit once the virus has been completely manufactured within the inside of the host cell. The N spikes will allow it to be released. And if this H and N sound sort of familiar to you when talking about flu viruses, it's because the H and N spikes are what are used to define the flu virus strains. So the specific array of proteins that are present on the surface of the virus's envelope dictates what its host range is, i.e., what organisms it can infect by entering those cells and exiting those cells. And so, as we can see, we have humans here. Humans can be infected by H1N1, H2N2, H3N2. There's a number of different strains that can infect humans. There are strains that can infect multiple different organisms. For example, H1N1 can infect both humans and swine. And then there's also avian flus, the bird flu, H5N1, that you've probably heard of before. So that's where the H's and N's come from. Those are the proteins that are on the surface of the flu virus, and they dictate what strain it is. So now let's take a look at how the flu virus carries out its life cycle by infecting human cells. So here we have in this depiction an influenza virus that is touching down and attaching to the surface of an epithelial cell. This is the cell that lines your respiratory system. So it attaches to your epithelial cell, and then the whole virus is engulfed. Unlike what we saw with bacteriophages, the bacteriophages injected their DNA into the inside of the host cell. Instead, in this case, the flu virus is engulfed via endocytosis, which we've spoken about endocytosis before. It's been a little while, but endocytosis is the process of engulfing and drawing inside materials to the cell by the formation of a vesicle, which is a little pocket in the membrane, which then captures the material outside of the cell, in this case the virus, and then seals off, engulfs it, and brings it inside of the cell. However, this is not the only way that animal viruses can enter host cells. They can also enter through something called fusion. And fusion is a type of entry that is possible for enveloped viruses, because remember, enveloped viruses have that extra layer of lipids around the outside of their capsid, and they can actually take that layer of lipids and they can fuse it or merge it with the cell membrane and then dump the capsid of the virus inside of the cell. Flu viruses can actually do both of these different entry methods, but that's not the case for all viruses. As you can see, non-enveloped viruses, it would not be possible for a non-enveloped virus or a naked virus to perform fusion because a naked virus lacks the envelope that would be necessary to fuse with the cell membrane. Okay, getting back to the life cycle, we can see now that the flu virus has entered the host cell, and now it's going to basically open up and release all of its viral contents. It releases its genetic material, which, remember, in this case is RNA, not DNA, and that RNA travels into the nucleus of your cells, your host cells, and it will replicate itself to make more copies of the virus's genetic material. In addition to that, the messenger RNA from the virus will be used to create more viral proteins, which are necessary to create additional capsids for the new viruses. And this leads us to another checkpoint where we're going to review some content that we've learned about in our last chapter. Which structure in the host cell does the virus need to hijack in order to complete this step? Or in other words, in order to turn the messenger RNA into proteins, which cell structure does the virus need to take over? And then finally, after the virus has created its nucleic acids and its proteins by taking over the host cell's enzymes in order to do so, the pieces of the virus are put together into new viral particles that are complete and whole, and they are ready to exit the cell. They do so, in this case, by budding off and not by killing the host cell. So this cell basically turns into a virus making factory. It lives on and continues to churn out new viruses, which bud off. The cell does not lyse and burst open as it releases the new viral particles. So now that we've taken a look in detail at how the flu virus replicates itself, I'd like you to think back to bacteriophages and name 2 ways in which the steps involved in the life cycle of the influenza virus are different from the steps that were involved in the life cycle of bacteriophages. So this concludes our look at how the influenza virus functions. Now we're going to move on to talking about coronaviruses. Coronaviruses have obviously been a hot button issue in recent years. The coronavirus, like the flu virus, is an enveloped RNA virus, meaning that it is a virus that has their capsid contained within an envelope, and as its genetic material, it has RNA rather than DNA. It's pretty small as far as viruses go. It's only about 0.5 microns in diameter, um, and it has these surface proteins that we've talked about that stick off around the edges and give it the appearance of a crown, which is why it is called the coronavirus. It enters and exits the body through the respiratory system, again, like the flu virus. In particular, it's transmitted through respiratory droplets, airborne particles, direct contact, or less commonly, indirect contact. Indirect contact would be something like you touch a door handle after a person who is infected with coronavirus has touched it and has shed some of their viral particles onto that door handle. Now, although we've been hearing about COVID-19, COVID-19 is not the only coronavirus out there. There are actually 7 known strains of coronavirus that have human hosts and many more that have non-human animal hosts. 4 of these are causes of the common cold, so very mild infections that largely affect the upper respiratory system rather than the lungs. However, there are a few very severe coronaviruses, including Middle East Respiratory Syndrome, Severe Acute Respiratory Syndrome, or SARS, and then finally COVID-19. How contagious is COVID? To give you a picture of how contagious any illness is, there is a quantity called R naught. That's what this R with a little subscript 0 stands for. You pronounce it R naught. This is a value that represents the number of individuals to which an infected person transmits the disease on average. So to give you some perspective, we have COVID-19, the Omicron variant, coming in with an R naught of about 3 to 3.3. This figure is actually outdated. This goes back to prior non-Omicron strains of COVID-19 that were less contagious. But what this means is that every person who's infected with the Omicron strain of COVID-19 gives it to somewhere between 3 and 3.3 other people on average. To give you some comparison, the R naught value for influenza is only 1.5, so about half as much as COVID-19, is about half as contagious. Measles, which is among the most infectious viruses known, has an R naught of 15, meaning that a person who has measles on average transmits it to 15 other people over the course of their infection. So that gives you a perspective of how contagious COVID is compared to some other illnesses. And as long as we're talking about COVID strains, we also should talk about mutations in the coronavirus spike proteins that have really driven the evolution of this virus and changes over the years as it has graduated from the alpha to the beta to the gamma to the delta and finally the Omicron strains. So this figure right here, I pulled from a paper that was published on the evolution of the coronavirus spike proteins. And it shows what this stands for. S1 subunit is spike protein number 1 subunit, spike protein number 2 subunit. What it shows in each little black part of the grid here is where a mutation has taken place in that particular strain, in that amino acid. So every box here is an amino acid or one little unit of the protein, the spike protein. And every time a box appears black, that means a mutation has occurred between the previous strain and that strain. As we can see for Omicron, lot of black boxes here. Lots of mutations have occurred in that spike protein that have made it more compatible with human cell receptors and better able to cause infections and more contagious. So, as you probably know, the Omicron variant is far more contagious than the Delta variant was, but it is less severe as well. Why is it less severe? Well, Omicron enters cells primarily via endocytosis, whereas Delta entered primarily via fusion. There's emerging research on this. And the upper airway cells have receptors on them in the human body that are more conducive to endocytosis compared to the lower respiratory system, which possibly explains the lower severity of Omicron, by which I mean Omicron symptoms tend to be more upper respiratory, ear, nose, throat type symptoms, as compared to Delta, which was far more lower respiratory, bronchial, lung, alveolar inflammation, pneumonia. And so this is possibly an explanation as to why Omicron is less severe. It targets more of the upper airway than Delta did in the lower airway. And I'm going to give you guys a checkpoint that looks closer at this published data. As I said, each box in the grid represents an amino acid. Each black box represents where there has been a mutation. And what these numbers and letters mean on the left-hand side of the figure is specifying what the mutation actually is. Because there is actually a code that biologists use that designates each amino acid as a single letter, which you can see here. This chart right here shows you all 20 amino acids, and they are categorized as polar, nonpolar, electrically charged, which we've talked about previously. And we can see in each red circle is their one letter abbreviation. So when we see, for example, L18F, what that is indicating to us is that a leucine, L, at the 18th position, the 18th spot in the protein, was transformed into a phenylalanine, an F. So that's what this mutation indicates. A leucine at spot 18 was transformed into a phenylalanine. Here, down here, it's where we see DEL. This means that the 69th amino acid was deleted entirely. That's what this deletion abbreviation means. So what I'd like you to tell me is how you would classify these mutations. Mutation number 1, this L18F, and mutation number 2, 69 deletion. How would you classify it? As a silent, missense, nonsense, or a frameshift mutation in each case? So now we have one more animal virus that we are going to be looking at. We've talked about the flu, we've talked about COVID. we are lastly going to look at human immunodeficiency virus, or HIV. So HIV is also an enveloped virus, and it is also an RNA virus where it has as its nucleic acid RNA instead of DNA. But HIV is also a sort of special case of a virus that has RNA as its genetic material because it has a enzyme called reverse transcriptase included within its structure that has the function of converting RNA into DNA. And reverse transcriptase is named very intuitively if we think back to what we've just learned in chapter 15, because we've learned that transcription is the process of the cell converting its DNA into RNA. So reverse transcription is the process of converting RNA into DNA. And that's what this enzyme reverse transcriptase does, because in the case of HIV, it starts with RNA and turns it into DNA. Now, what is the significance of this enzyme in sort of the grand scheme of HIV's life cycle? We'll talk about that in just a minute. But as you probably know, HIV is a virus that affects the immune system, specifically by infecting and killing human T cells. T cells are the type of white blood cell that we have that activates our immune system. So, as a person has an HIV infection that goes on and is untreated, it can eventually deteriorate the body's immune system and lead to the onset of acquired immunodeficiency syndrome, or AIDS. So, HIV is not the same thing as AIDS. HIV is a virus that, when it infects a person and that infection goes untreated, it can progress to AIDS. However, if treated, then very often the infection does not progress and turn into AIDS. So this figure right here, which I realize is a pretty busy figure, so I'm going to walk you through it, shows us exactly how HIV carries out its process of infection and replication. Again, we have the same basic series of steps that we saw with other animal viruses and with bacteriophages, but there's going to be a hitch in the middle where that special enzyme reverse transcriptase comes into play. So first of all, the HIV virus attaches to the surface of host cells. Remember, these are T cells, human T cells, part of the immune system. And after it attaches, it enters, releases its viral RNA, and then the reverse transcriptase enzyme, which is represented here by this little green sphere, will take the virus's RNA and it will reverse transcribe it into DNA. That DNA then integrates itself into the DNA of the T cell. So, in other words, it joins with your DNA in your T cells, it inserts itself. Now it is there, and it is there permanently. So this is why HIV infections are a lifelong infection. The DNA of the virus inserts itself into your DNA. It becomes part of your genetic material. And then, again, your cell will use its own machinery, its own enzymes and resources to replicate the HIV RNA, turn it into HIV proteins, assemble new viruses. Sorry, I'm a little bit behind here. Assemble new viruses, and then eventually release them. And like with the flu virus, the release here happens without killing the host cell immediately. Although over time, as these T cells are turned into HIV making virus factories, eventually the T cells do die over the course of being made to produce HIV viruses. So now learning about how HIV replicates, I'd like you to tell me in this checkpoint what HIV has in common with a bacteriophage that carries out the lysogenic cycle. What does the lysogenic cycle and the life cycle of HIV have in common? Okay, so in the last part of this lecture, we are going to just briefly talk about how viral infections are prevented and treated. Because, like I said at the beginning of this lecture, Antibiotics are not effective against viruses. Antibiotics are classes of drugs that are designed to fight cellular organisms, living bacterial infections, and viruses are not that. Viruses are particularly difficult to treat because they enter your cells and use your resources and your enzymes. And so killing them requires some really clever thinking about how we can attack the viruses without also attacking your cells. There are, however, many vaccines that can prevent viral infections. And there are some specially targeted antiviral drugs that are available to treat certain particularly bad viruses. And we'll talk about a few of those as well. So first, let's take a look at the flu vaccine. The flu vaccine is a unique vaccine because, unlike other vaccines that are administered on a determined schedule, a limited number of times throughout childhood or adulthood, the flu vaccine is updated every year. And that is because the spike proteins on the surface of the flu virus mutate at a very rapid rate. And so by the time the next season comes around, the previous season's vaccine no longer offers protection against the strains that were dominant in the prior season. So, new vaccines are developed every flu season, but sometimes the mutations move faster than vaccine development, which results in the vaccine that is developed for an upcoming season actually being a bad match for the flu that circulates that year. This is what happened in 2017-2018 flu season, which was the worst season on record since they started doing epidemiological surveillance of flu. 2017-2018 was the worst season ever recorded. And one of the reasons why is because over 90% of the H3N2 flu vaccine that was included in that year's vaccination was not protected against. The strain had evolved and it had mutations that made it a bad match for what was included in the vaccine. And as a result, over 60,000 people in the US died from the flu during that 2017 and 2018 season, which is a much higher number than typically die from the flu every year. And we can see in this figure right here from the CDC that there is a huge spike here in the 2018 influenza deaths compared to the years on either side of it. Another reason why the 17-18 season was particularly bad is because the strain H3N2 is particularly difficult to protect against. Seasons where this is the dominant strain that circulates are historically bad for a couple of reasons. First of all, H3N2 just seems to mutate more often in ways that make it a bad match for the prepared vaccine. But also, H3N2 is more vulnerable to something called egg-adapted changes because our flu vaccine is actually grown inside of chicken eggs. In order to create the vaccine, you have to create a large number of flu viruses, and those flu viruses, being viruses that require a cell in order to replicate, have to be grown in a living organism. And a natural choice is chicken eggs, given how large they are, how abundant they are, and how easy they are to work with. And so this is how we produce our flu vaccines. However, the egg environment is obviously different than a human. Chickens are different genetically than humans. And so when the flu virus is grown inside of the chicken egg, something called egg adapted changes occur to it. And this can further cause mutations that make it a bad match for the flu that is circulating among the population. Now, this brings us to another checkpoint pertaining to flu, not pertaining to flu vaccine and flu prevention, but flu treatment. Because there are also medications out there that can treat flu infections once they have already taken hold, and the most common of these is Tamiflu. Tamiflu is a med that can shorten the duration of a flu infection and reduce the severity of symptoms. It's not a cure, but it makes the duration of your infection shorter and it makes your symptoms better. And it does this by inhibiting the N spikes, or the neuraminidase protein spikes that are on the surface of the flu virus envelope. So I'd like you to tell me which step of the flu virus life cycle does Tamiflu interfere with? So we have talked about the flu vaccine. What about vaccines that protect against COVID-19? There are a number of vaccines that have been developed for COVID-19, and they have two different formulations as of the recording of this lecture. The vaccines developed by Pfizer, Moderna, and Johnson & Johnson all contain a piece of messenger RNA or mRNA that encodes the coronavirus spike proteins. So all your immune system really needs in order to gain protection against a virus is to recognize one piece of it. Oftentimes that is the spike proteins that if your immune system gets to know what those spike proteins look like, it develops a memory of them, and then it can mount an attack on the virus if it encounters it in the future so that you don't become ill. So these vaccines, they don't contain the coronavirus itself. They don't even contain the spike protein. They contain a fragment of mRNA that has instructions for how to make that protein. And so what happens when this vaccine is introduced to your body is your cells will take that messenger RNA, they will synthesize the spike protein using the instructions in the messenger RNA, and then based on recognizing that protein, they will develop an immune system memory of what it looks like and be able to attack the COVID virus in the future. And on the next slide, I'll show you just a little bit more detail how that works. But there's also another vaccine available at this point from the company Novavax. The Novavax vaccine contains the spike protein itself. Rather than containing instructions for creating the spike protein, it actually contains the coronavirus spike protein. So it's a little bit more of a direct route for administering this immunity to your body. But this figure right here, which again I know is a really busy figure, sort of shows you what happens when a mRNA vaccine like Pfizer, Moderna, and Johnson & Johnson formulations are delivered to your body, what happens inside of your body. So right here up in the left, we see the messenger RNA that is found within the vaccine. It gets accepted by your cells, and your cells will then take that messenger RNA, run it through the ribosomes, and they will produce the spike protein, which is shown here, green and yellow. It will produce the spike protein and then take those coronavirus spike proteins and move them to the outside of the accepting cell, whereupon another cell from your immune system called a B cell comes along, recognizes that spike protein as a foreign element, and it will then create antibodies that are able to recognize it and circulate those in your bloodstream so that you have this permanent immunological memory of what that spike protein looks like, and you have antibodies that are ready to go on the attack as soon as a COVID virus is introduced to the body. Lastly, we are going to say a few words about what is actually the largest class of antiretroviral or antiviral medications in existence, and that is antiretroviral medications or medications to treat HIV. There are actually more medications out there to treat HIV, this one singular virus, than there are all medications that are used to treat fungal infections. So there are quite a few HIV antiretroviral medications. And the way that these medications work is by targeting one of the HIV virus's few enzymes. Some of the more common classes of these drugs are reverse transcriptase inhibitors, which disable the enzyme reverse transcriptase, which we've talked about. And in doing so, they prevent the virus from converting its RNA genetic information into DNA, and they put a stop to its life cycle. Integrase inhibitors disable an enzyme called integrase, which is responsible for taking the viral DNA and inserting it into the human chromosome. So again, they put a stop to the life cycle, but they do it at a different point than the reverse transcriptase inhibitors. And then lastly, the protease inhibitors disable an enzyme called protease. And this enzyme is responsible for assembling new viral pieces that are created into a complete virus. So they shut down the process at that stage. Now, these medications are taken if a person has an active HIV infection, but they are also highly effective when used preventatively or prophylactically. So if a person is in a position where they are at a known higher risk for acquiring HIV, then they can take these medications in what is known as pre-exposure prophylaxis, or PrEP. So people can pursue PrEP, for example, if they have a partner who has HIV, if they use intravenous drugs and share needles and they know that they're at a higher risk. These medications taken prophylactically are extremely effective. They're 99% effective at preventing an HIV infection from taking hold in the first place. And they're also very effective at staving off the progress of an HIV infection and preventing it from progressing to the point where it becomes AIDS, acquired immunodeficiency syndrome. So these data, keep in mind, these data that I have presented here are from Canada. So Canada is a rich first world country where there's very good access to these types of medications. And throughout many parts of the world, in developing areas of the world, there's still very high death rates associated with HIV. However, when there is access to antiretroviral medications, the life expectancy of a person who is HIV positive is actually not that much lower than the life expectancy of a person who is not HIV positive. So, for example, we can see in the range of 2008 to 2012, life expectancy for a person who's HIV positive in Canada, 72.4 years. It's been going up steadily over time. And we can also see the effect of ART, antiretroviral therapy, on life expectancy at the point where it was introduced, or HIV-related deaths, rather. HIV-related deaths had increased steadily throughout the late 80s and 90s. And as soon as these drugs became available, you can see how many lives they have saved over the years, the combination of these various drugs for treating HIV infections. One factor that is becoming problematic in HIV infections is, however, drug resistance. And drug resistance in HIV emerges very quickly because reverse transcriptase, this enzyme, makes a lot more mistakes than a regular DNA polymerase enzyme. When it creates DNA, it makes a lot of errors, and as a result, retroviruses like HIV have a very high mutation rate. And because of those mutations, they are able to evolve resistance to drugs much faster than even bacteria do. And so, for example, it is now common for HIV strains to be resistant to the very first antiretroviral drug that was developed in 1987, which is azithromycin, a reverse transcriptase inhibitor. And we've now characterized exactly what that looks like. This right here is their depiction of the reverse transcriptase enzyme. And normally, the way that this reverse transcriptase inhibitor works is it attaches to reverse transcriptase down here, and it blocks it from performing its job. However, reverse transcriptase now has evolved an extra component to the protein, which essentially blocks the drug from attaching to it any longer. So drug resistance is a problem. However, we're always adapting new ways to administer these drugs, and new versions of them. Recently, the WHO recommended and approved a long-acting version of these pre-exposure prophylactic medications, which are able to be administered on a much more infrequent basis and using an injection. So rather than taking an oral pill every day, people are able to get an injection of these drugs once every few months. And it is not as effective as oral everyday medication, but it's about 79% effective. And that is pretty good for parts of the world where there is, for example, not a medical clinic accessible close by, and people can't get to a clinic on a daily or even weekly basis to access medical services. So there is hope, and there's continuing incremental improvements in the fight against HIV. Okay, that's all I have for you on viruses. So congratulations on completing your last lecture of the semester. I hope you learned a lot. And if you found this interesting, then you are going to love microbiology, Bio 205, and I hope to see some of you in that class.